We'd like to welcome everybody back to the 2025 Speaker Series celebrating Autoimmune Encephalitis Awareness Month. The topic is appropriate today, given that today is Rare Disease Day as well. Why do we need to study the genetics of autoimmune encephalitis? My name is Tabitha Orth. I'm the Executive Director and founder of the International Autoimmune Encephalitis Society. And before we get started, I just want to point out that at the end of the presentation, we'll be doing a question and answer period as time allows. And if you go to the bottom of your screen, you'll see a Q&A box there. So as questions arise, make sure you form them as a general question, not a case-specific question. A bit of housekeeping. The recording of this webinar will be uploaded to our YouTube channel later today. We recommend you subscribe to our channel, like and click the notification bell so your alert is uploaded. And today we're very fortunate to have Professor Emmanuel Mignot with us. Professor Mignot's research group at Stanford University in California is currently conducting the IGNITE study, which investigates whether patients diagnosed with autoimmune encephalitis have a genetic predisposition to AE. He is the Craig Reynolds Professor of Sleep Medicine in the Department of Psychiatry and Behavioral Sciences at Stanford University and the Director of the Stanford Center of Narcolepsy, and recognized as having discovered the cause of narcolepsy. Professor Mignot's practiced medicine and psychiatry in France for several years before serving as a visiting scholar at the Stanford Sleep Disorder Clinic and Research Center. He joined as faculty and director of the Center of Narcolepsy in 1993. He was named professor of psychiatry in 2001. He has received numerous awards for his work, including a 2023 Breakthrough Prize in Life Sciences, and is a member of both the National Academies of Sciences and Medicine. Most of his current research focuses on neurobiology, genetics, and immunology of narcolepsy, with indirect interest in the neuroimmunology of other brain disorders such as autoimmune encephalitis or neurological paraneoplastic syndromes. And with that, I'm going to turn it over to you, Professor.
Thank you so much, Tabitha. Can you all hear me well?
Yes, we can.
Okay, so I'm going to share my slide here. So today I'm going to talk to you about the genetics of autoimmune encephalitis and why it is important and what we are starting to find, and give you some directions on how this is going to evolve and maybe to the benefit of patients.
Hello.
So, yeah, can you hear me? Okay, so first, you may wonder why we need to study the genetics of autoimmune encephalitis. So the first reason I think is that, many of those diseases have a strong genetic component, and as all autoimmune disorders have. The second reason is that every autoimmune disorder is different. Like people have studied narcolepsy, type 1 diabetes, multiple sclerosis, celiac disease. And when you study each of these disorders, they are slightly different. They have a different cause, and different subparts of the immune system are involved. And it's very important to determine what type of subpart of the immune system is involved in each disease, because this way you can actually target each disease differently with better treatment. One other big advantage of genetics, which people don't really appreciate, is that when you find a genetic factors that's associated with a disease, the big advantage is that it's always causal. So what I mean by that is when you have a disease, let's say anti-NMDA receptor encephalitis, and you find a genetic factor that's associated with it. The genetic factor has to be involved in causing the disease because you cannot modify your gene from having the disease. You are born with them, so they have to be on the bottom of the causality pathway, which is very different if you study blood cells, immune cells, if you study things in the blood, you can find something that's associated with the disease, but you never know 100% if it's the cause of the disease or if it's a consequence of the disease. You could very well have a disease that then changes the immune system in a certain way, but it's really not critical to the cause of the disease. So the genetics has a big advantage is that it's always causal. And as I will discuss, one of the essential genetic components of this genetic autoimmune disease is a special system that's called the HLA or MHC, human leukocyte antigen, or MHC, major histocompatibility complex system, which is a series of genes that are very complicated, located on chromosome 6, and that are very important for regulating the genetics of the immune system in different people. They are very important for all kinds of diseases. Actually, there's probably about 200 or 300 diseases that are associated with different genetic markers in the HLA. So this is really crucial to most autoimmune diseases. What's important also for me is that relatively little has been done in this general area of genetics of autoimmune encephalitis. Many of the diseases now have been studied genetically. The tools are very, very powerful. And yet, I think autoimmune encephalitis is relatively new. Very few people have done work in this area. So the main things that we can find maybe by studying the genetics of autoimmune encephalitis, first, it could help the diagnosis. Sometimes we might be able to find a genetic markers that help to confirm that someone has this problem and not this problem. We may find that certain genetic markers are going to help to predict how severe is the disease, how it's going to evolve. But most importantly, at the end, maybe it's really finding the root cause of the disorder and then designing special therapeutics once we really understand which arm of the immune system is the one that is being impacted by each disease. So you all know that there have been an incredible growth of this area, for a long time, and it's difficult to believe, but in the 1960s or 70s, many people thought that the brain was protected against autoimmune disease. And in fact, many mistakes were done, in the past. Like many people thought that the immune system was also not important for cancer. Yeah, it's unbelievable, but some people had done had studied mouse and had grafted cancer, and some of these mouse had no immune system that were nude mice, and they didn't see that the cancer was growing faster in the normal mice versus the nude mice. So they say, oh, the immune system is not important for cancer, which we know is completely wrong. And the same way, many people at the time, they had discovered many autoimmune disease, but very few affected the brain. So for a very long time, people thought that autoimmune disease, the brain was protected from the immune system, that somehow, the brain was sealed from immune cells to go inside, which is completely wrong. And we know now that there is very high regulation of cells going into the brain, and they can attack neurons, they can attack glial cells. Of course, there is multiple sclerosis, which was known for a long time, but was a little particular because it affects more the conduction sheath of the nerves, the myelin sheath, not really the neurons itself. But otherwise, for the brain, a lot of people thought that it was relatively rare. But clearly, that has been an explosion. Initially, the first autoimmune disease of the brain that were discovered that maybe had this autoimmune encephalitis profile were discovered in association with cancer. That's why a big area or people who have been starting to work in this area, they come from two different directions. One of them is really a lot of people who worked on cancer and discovered that sometimes when you have a cancer, you develop an immune reaction against the cancer that becomes, as I told you, this idea that the immune system was not important for cancer was completely a fallacy. Because clearly, in a lot of cases, when you have a cancer, often you have another symptom that's independent of cancer. It's actually very common. People, when they start a cancer, sometimes they know something is wrong. They have a rash. There's something else going on. Low white blood cells, red blood cells. And sometimes it's because the immune system is trying to get rid of the cancer. And by attacking the cancer, it may also cross-react with something else in your body because your cancer cells are very close to your normal cells. And then it starts to cross-react with something and you have another autoimmune. It's very common. It's called a paraneoplastic syndrome. Para means on the side of, and neoplastic is cancer. So it's basically on the side of the cancer. It's when people develop specific cancer, sometimes they have other diseases which are related to the fact the immune system attacks the cancer, and by attacking the cancer, it also cross-reacts with other parts of your body, and you have some weird disorder in association with it. And generally, it's believed to be really mostly due to when you have a cancer, you suddenly start to express a lot of things in the cancer cell that may be a bit abnormal and at very high level. And because there is so much of it, the immune system realizes it's not normal and start to try to attack it. And then it starts to cross-react with your own self in some ways. So this has been known that the first autoimmune disease of the brain were discovered a lot in the context of this cancer. But then later on, people discovered that a lot of these diseases, autoimmune encephalitis in particular, were sometimes not associated only with cancer. So it became that some diseases were associated with cancer as a consequence of the cancer immune system reactivity against the cancer, but then sometimes there were no cancer associated with it, but you had the same disease. So somehow you could have another mechanism that can create the same immune and normal response. And some of them were then discovered even to be without any cancer. And these were more regular autoimmune diseases. So why is that important to study the genetics? It's also important to study the genetics because it's going to help you to understand which aspect of the immune system is involved in these different diseases. For example, the immune system as generally, we consider three different main arms. In fact, it's not true, it's much more complicated, but there's three main systems that are very important for the immune system. One of them is the B cells, which secrete antibodies, and the antibodies are floating in the body, and they basically fix virus or bacteria. And then there's another system that gets involved and helps to clear up the virus and bacteria through the antibodies. So the antibodies is a very powerful way of fighting bacteria, virus, and of course, it's also one of the ways that the immune system can attack your own cells. And a lot of diseases are associated with autoantibodies, antibodies that instead of recognizing viruses, they recognize a piece of your own cell. And antibodies are a great disease marker because they're very easy to measure. You can measure them in blood, and we have very good technology to measure them at very low level. So it's a great diagnostic marker, but it doesn't always mean that it's causal to the disease. Maybe it's just because there is an immune reaction, and the immune reaction has resulted to the production of these antibodies that target something, but it's not so important to cause the symptoms. But still, they can be great disease markers. Then there is another system, which is the CD8 cells, or called cytotoxic T cells, which are more involved in killing directly cells that are infected with a virus, and they can also kill cells from a tumor or kill normal cells in an autoimmune disease. So these are the two main arms of the immune system, either the B cells that produce antibodies or the T cells that sometimes can really attack directly the cells that are infected with the virus or cells of your own self in an autoimmune disease. And there's a third population of cells that's also called CD4 cells, which are very important because they're kind of the general of the immune system. That's how I like to call them. It's kind of orchestrate everything. So they are pretty much always involved because they are the first line of defense that decide, oh, should you produce antibody or should you rather make cells that are going to kill cells that are infected or part of your tissue. So they are the ones that really decide, is that more like the cell-mediated immune system that's going to be activated, or the B cells, or both. And often both of them are activated, but in all autoimmune disease, everyone discusses which one of these two arms is the most important, because the treatment is going to be different. We have treatments that can deplete B cells that are very active in antibody-mediated cells, but we have much less good treatment for treating disease that are involving CD8 cells that directly destroy the tissue. So one other, like, very brief, in fact, it's a complicated slide, but I'm going to summarize it very quickly. How does it work? So the key with the immune system is quite amazing, is that it's very specific. So it can recognize things in your body, or viruses, or bacteria in a very unique way. And its specificity, it all comes mostly from this particular molecule that are called HLA or MHC, which I will mention are very important genetic factor in the immune system and autoimmune disease. And this HLA, I also represented them here. There's two flavors. There's one that's called class 1 and one that's called class 2.
We actually are not seeing your slides.
Oh my God, you should have.
I'm sorry, I was waiting when you referenced it. I thought, well, maybe he's.
Okay, oh, I'm sorry. Then
That's all right, that's all right.
Very difficult to follow what I was saying.
I was actually following it okay.
Okay.
There we are.
I'm sorry.
That's all
must have
right.
been very difficult because you have the B cells here, as I mentioned, and these are the CD8 cells, which are more like the ones that are, we call them cytotoxic T cells. They kill directly the tissue, basically, or the virally infected cells. And this one are the CD4 cells, which are the general of the immune system, kind of. They are just decide, is it going to be B cells that are going to produce antibodies, which are going to target, the tissue or viruses, or is it going to be more like a CD8 cells that are going to attack the tissue? So these are the two arms of the immune system. But the key component that regulate what it recognizes, and that's very important. This is just a general theme, but the immune system was very particular. It's going to recognize a particular tissue. It may attack, the cells of your brain, a very small group of cells. And one of the questions is, how does it do that? How can it recognize a very specific, I should probably here. How does it recognize specific tissue? Why in autoimmune encephalitis, like anti-LGI1, it recognizes certain group of cells in the brain, and suddenly you have seizure, whereas in others, you have a completely different set of symptoms. The reason is that the specificity of the immune system starts with this HLA molecule, which I started to mention, which are these molecules that are expressed on the surface of cells that are pretty much everywhere in the body. And they have a very specific area here that is very that can bind pieces of virus or antigens or your own cell in a very, very particular way. And then it produces an immune response that is going to target this particular protein. So it basically recognizes a certain piece and then targets that protein by recognizing it through the HLA molecule, either class 2 or class 1. And class 1 is more when it kills the cell, and class 2 is more for orchestrating the whole immune response. So this HLA molecule here they are very particular because they are critical piece of the immune system that help to understand why the immune system can recognize one particular tissue. So, for example, if you have an autoimmune disease that targets the thyroid, it means that there's probably a piece of thyroid hormone or thyroid protein that is recognized by a particular HLA class 2 and then triggers a whole immune cascade that recruits these B cells to create antibody against thyroid, and then maybe T cells that are going to kill the thyroid cell because of recognizing this particular piece. So it's really important. I know it's a little bit complicated, but that part I think is really important to just generally understand. It's the way the immune system recognizes yourself, your non-self, or virus or bacteria is through these particular molecules that are called HLA or MHC. And the way it works is, again, these MHC, these HLA molecules, they are very different in different people. So, for example, that's why, in fact, they were first recognized in transplantation. So, for example, the HLA, my HLA, I know, I mean, I have many different HLA genes, but one particular one, I know that I'm DR 1301 and DQ and DR 701. And this is very mysterious nomenclature that reflects the fact that in my HLA molecule have a little different amino acid, different structure in these A, B, C, D, E, F pockets here. And these pockets is what binds pieces of yourself, for example, LGI1 or NMDA receptor or virus. And then this is being recognized by another set of cells, which are the T cells, that then, as I explained here before, are going to orchestrate this immune response. So this first step here is super important. It's how the immune system recognizes what it wants to create an immune response against. And what is very unique about it is that these HLA molecules are different from one person to the next. So most of the genes that we have in our body, we all have the same genes, because let's say that we want to produce glucose. Of course, we are all are going to have a gene that are going to metabolize glucose. And for everyone, the gene that metabolize glucose is going to be the same gene, and it has very little differences between different individuals, because if you create a difference, it would not work. But here, it's a completely different system, where actually there is design differences between people. These HLA genes or molecules are very different from one person to the next. So, for example, I'm sure, Tabitha, you will have completely different HLA polymorphism. And the reason is that this way, if you encounter a virus, like, for example, COVID, you will probably bind a certain piece of the coronavirus in a different way as me, because you will have a different HLA subtype, which will bind differently the piece of the coronavirus. And then this way, you will develop an immune response against the coronavirus directed against a certain piece of the virus, whereas I will bind another piece because I have slightly different sequence here, and I may develop an immune reaction against another piece of the virus. So this way, this HLA is really incredible because it allows to individualize the immune response. So everyone, when we meet a virus, a bacteria, or we have an autoimmune disease, we have these HLA molecules that allows to direct the immune response against certain pieces of the protein of the virus in a different way. So that's why these genes that produce this particular molecule are very important to the general regulation of the immune system. And many, many different diseases are associated with variants of these different HLA genes. And I'm sorry, because it is a little bit of a nightmare, because these genes have a very strange nomenclature, So these HLA gene or MHC genes, they are all located on one particular chromosome that's called chromosome 6. And there are two groups. There is a class 1, which are very important for the recognition by CD8 cells, cytotoxic cells. And then there is a class 2 HLA molecules, which are very important for the CD4 molecule, CD4 cells. And you see here is like the nomenclature is very complicated. This is the most complicated part of my talk, by the way. Is that these HLAs, they are usually noted like this. You have one type of gene like A, B, C, D, R, D, Q. And then after that, you have a broad subtype. So O1, O2, O3, which is like a family of the sequence. And then O1, a second that makes a slightly different subtype. For example, 301 and 303 will be almost the same. So basically, that's the way it is. It is all my subtypes are denoted like this, like HLA-DR and O1, 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 O1 means it's a broad subtype O1 with a very specific subtype O1. And it has very specific amino acid here, which correspond to the pocket here that will bind the little peptide that I told you will trigger the immune response. So it's really how your particular immune system is going to recognize one particular piece of your body or virus to produce an immune response that's specific for that virus or that protein. And you see that all these subtypes here, they have certain regions of the molecules where they are different, and that actually corresponds to this pocket where it actually puts a piece of the antigen, the protein, the piece of the virus or your cell that then allows the immune response to be different in different people. So I know it's very complicated, but it was important to mention this because this HLA system, you may have heard about it, but a lot of people don't understand it. And very justifiably, it's normal because it's one of the nightmares of genetics. It's the most complex series of genes that exist in the body, in the sense that they are very different from one person to the next. And they really regulate how your immune system is your immune system, not the immune system of anybody else. So obviously, they're going to be very important to predispose to all kinds of diseases that involve the immune system. And in fact, these HLA molecules are the cornerstone of the predisposition to a lot of infectious disease and autoimmune disease, in particular, a lot of autoimmune encephalitis. But the second thing I wanted to introduce is how do you get to a genetic base of the disease? So there's two methods. One of them is to study family of people with a disease, but family don't work very well when the disease is complicated. And we know that for autoimmune encephalitis, I'm sure you know that most cases, they are not familial. So it's not like there is a pure mutation. It's more an accumulation of many things that occur that produce the disease. And in these cases, the best way to try to study which genetic factor are involved is to use a method that's called genome-wide association study. So basically, you have these chromosomes, and now there have been genetic markers that have been sequenced. So we know pretty much a sequence of every chromosome. And we also know how many genetic variants there is all through the chromosome between different people. So regularly, all through the chromosome, there is often one particular position where some people have a G and another have an A. And by the way, in chromosome 6, where there is HLA, there is a lot of these changes, abnormally high. That's why it's so different from one person to the next. But in general, for the rest of the genome, every 1,000 base pair, there is one change. So now what you can do is you can actually study 1 million different variants on all the chromosomes, and then you can see if any of them is different between the cases or the controls, and then it tells you that here there must be a gene that predisposed to a particular disease like autoimmune encephalitis. So that's called a genome-wide association study. And what's strange is This has not been done that much in, it has been done in everything. It has been done in high blood pressure, height, diabetes, heart disease, narcolepsy. But honestly, for autoimmune encephalitis, nobody has done a lot of work. So it's a completely virgin territory. There's everything to discover. So with that technique, you can kind of scan all the chromosomes and try to find all the genetic factors that are actually increasing the risk of having the disease, including HLA, of course. And as I mentioned at the beginning, what's really useful is that these genetic factors are always causal. So by studying what they do, you're going to get an idea which part of the immune system is really important for the disease. And maybe, as I will explain, especially with HLA study, what is really triggering the beginning of the problem that leads to this abnormal immune response. So I will give you a few examples of diseases that we have studied and what we are finding, and that will give you an idea of how that can be applied, because otherwise I think it becomes quite technical. So we will take one example, is this anti-LGI1 encephalitis. I'm sure some of you know this disease. It's a disease where people have these seizures that are very particular, where it's called facial-brachial dystonic seizures, where people have a strange movement. And it's quite common, and sometimes people have a psychiatric problem and changes in electrolytes. And the big problem is that a lot of these people, when they recover, they are not completely normal. They still remain having a chronic problem, and they never completely recover. So we really need to understand quickly what is the cause of it and intervene very quickly. And we know that they have antibodies against these proteins that's called LGI1, which, to be honest, we don't really understand what it does. We just know that this protein is expressed everywhere in the brain. And for a reason that we don't understand either, there is one particular region that seems to be more inflamed, where somehow the immune system seems to attack this LGI1 or other proteins that produce these symptoms. So when you do a genetic, like I was trying to explain, this is what's called the GWAS. So that's what I was trying to explain initially. Each of these blocks here, represent a chromosome. Chromosome 1, 2, 3, and you see we have 22 chromosomes. And then each of these dots is one of these genetic markers that I showed you here. So, and each of them is basically a probability. So it's what is the probability of this genetic marker to be different. I don't know why it's behaving like this, to be different between a case and a control. So for example, here you see the problem here is we are studying 1 million different markers. I mean, it's a huge number, right? So of course, you cannot just say, you have to go to a very, very high probability for this not to occur by chance, right? Because when you study 1 million markers, you will always find something that's different, right? So that's why for this genetic association study, you need to have a lot of people. You know, the minimum is usually 300, but practically you almost need 1,000. Otherwise, it's quite difficult. But when you're lucky, even with a few people, you can find something. And for example, if you do this in LGI1 and you do like about 300 patients, you see that there is this big peak here, is that right? And this corresponds to one particular genetic region of chromosome 6, where there's a lot of little markers that are different between control and cases. And you see the p-values go to 10 minus 10, 0.000000, with 10 zeros and 1. So obviously it's extremely significant, and you can even divide by 1 million markers, it's still, you know, very significant. So we know, and this region is the HLA. So for example, when you just do a survey of this disease, you immediately see that the main genetic factors that predispose is one particular HLA. which I told you is this very complex immune response gene that is associated with a lot of autoimmune disease. And indeed, we found that it was found by someone else actually, that it's this particular subtype of 0701. And recently, this was known before I even started to work on it, okay? And people now, we have done some more work, and we figured out that if you have 2 doses of it, it increases even more the risk. And we also found this additional other genetic variant of HLA, which also predisposed to anti-LGI1. So we found that there is this subtype, which is a main subtype. You almost have to have it to have LGI1. And if you don't have it, you often have this one. Initially, actually, people didn't believe our finding, but then it was interesting because there was mostly Germany, they didn't believe our finding. But actually, we discovered that the main reason is that it's mostly associated with the Jewish ancestry, and they don't have a lot of Jewish people in Germany, unfortunately. So in fact, that's why it was not in Germany, but in the US, for example, you find it, and in Israel, it's very associated with this subtype. So now we know that there are actually 2 different HLA variants that predispose to autoimmune encephalitis. One is this variant of HLA, and one is this variant of HLA. And we found also that there is different, it's also, the presentation is different. For example, the people who have this particular HLA, they are always younger, so, and more severe. So that's just, not that important, but it's just to show you that not only it helps the diagnosis, but it also can help to understand maybe why is it more severe, and probably we're trying to figure that out. So it was confirmed actually in the Jewish population. It's the most common associated HLA for anti-LGI1 encephalitis. So the next question is you try to figure out how does it work. Now we know there is 701 and there is 402 that seems to be predisposing to the disease. It's talking to me. And basically you align the sequence and you try to figure out how it binds the same piece of the LGI1. And that's what we're doing here, for example. We're kind of comparing what type of structure the 701 binds. I don't know what's happening. It's really annoying. Versus this particular subtypes that predispose to LGI1 encephalitis, like the 402. So the next thing that we do when you discover that, you can screen the pieces of LGI1 and you try to figure out which piece can bind this HLA, which I told you is this pocket, 402 and 701 in a similar way, and maybe produce the same reaction that at the end is going to produce the autoimmune process. Because then eventually we might be able to block specifically this very specific reaction and prevent the disease or even treat it, but very specifically without touching any other arm of the immune system. So another example is this anti-NMDAR disease, which is a more recent disease that was discovered. This one is never associated with cancer. Like actually LGI1, it's also not associated with cancer. And it's a very, very terrible disease. It affects people who are relatively older, and then you develop some kind of dementia. And you have problems with your sleep, and then you also have problem with your balance, and then at the end, your whole brain goes. It's really a terrible disease. And it was discovered that there is some neurodegeneration. So for example, here, you see you get this tau, which is a sign of neurodegeneration that you find in Parkinson's disease that seems to accumulate certain parts of the brain. So somehow there is antibodies against this protein called IGLON5, which again is everywhere in the brain. We don't know why it seems to impact more certain region and not other region of the brain, but somehow it produced later a neurodegenerative disease. And this, we did the same thing. So you see again, this is the GWAS. And you see again, the main factor is again the HLA. We actually are finding some other things, but it's still a bit too early. And we now have about 150 patients, which is still low. You have to be very patient. Every patient I try to get a blood sample because you need large samples to figure out this kind of study. And here what we found was a little different. We discovered that it's actually 3 subtypes that seems to all bind and predispose. They are just very similar to each other. That's why they have the same digit, you know, 0105 or 501 or 0101, almost the same, or 104, almost the same. So it's really this combination of factors that binds a certain peptide that then produces the immune reaction that eventually allows the development of this disease. And we also found that it changes the clinical picture. You know, depending on the subtype you have, you might be more severe or not. So it's not only its different predisposition, but it also changed how early you start the disease. A little bit like with the LGI1, where we found that different HLA subtypes give a slightly different evolution. So they are slightly different subsets of the disease, but they still probably work the same way. It's just the HLA binds slightly differently, the peptide, and triggers a little bit of a different immune response, more or less severe. And all this, I mean, they're very similar subtypes, you know, so that's why it's very clear. And we can predict what kind of peptide should be binding this particular molecule that predispose to anti-GLON5. So now, of course, we are screening the GLON5 protein to find the peptides that bind it. And then you can complex them with the HLA, and you can try to screen T cells to find what T cells are abnormally reactive to this piece of peptide bound by the HLA that predisposed to the disease. So that's what we are doing for these diseases. So I think I'm going to pass on T-caspity because that's another one that's very similar. But one of the diseases that we work a lot on is anti-NMDA receptor encephalitis. And the reason is that it's different from the other one I discussed. When you do a genetic scan, you will see that you don't actually find the HLA, which is a little bit of a surprise because HLA is involved in all of this autoimmune disease. But as you will see, we usually don't find HLA as a main factor. So this disease, you all know, it has antibodies against the NMDA receptor, which is a very important receptor for a chemical called glutamate. And glutamate can be blocked by ketamine, which some people may have heard of. But ketamine is an anesthetic agent that also makes people crazy. So basically, these people is a little bit like they were flooded with ketamine, so they experience some kind of psychosis and seizures. And then, thankfully, sometimes people discover it's not schizophrenia, and then, but most of the time, you know, when people develop seizures, and then it's treated properly. But sometimes it takes time to be diagnosed, which is a big problem. And initially, it was found to be associated with a tumor. So more the second type, the ones that are paraneoplastic. And but now it's only about 10% that are associated with a tumor. And this one, we have really done a lot of work because we have a lot of patients. The anti-LGI1, you know, we are a group of people, so we're trying to do things in coordination, you know, because there's so few people working on these diseases. I mean, there's no point in competing. We're helping each other. So I took, you know, this disease more particularly, but the German group, they're working more on the anti-LGI1, for example. So we discovered something very interesting for this particular disease. As you see, the HLA, which normally is very high in all of these diseases, because it's very important for a lot of autoimmune disease, somehow there is a signal, but it's very weak. But one that there is a very, very strong signal in a gene that we never heard of before, that's called ACP2, and another one in another gene that we never heard of before, that's called DLMX2. So I will go through this. What was very surprising here is that this signal, especially the ACP2, was completely absent in all other ethnic groups. So we compare Caucasian, Asians, African, and this signal here is only in Caucasian. So it's very odd, you see. And we discovered something very bizarre. Is actually everyone that has this genetic factor, you have to have 2 copies of it to be susceptible. And everyone that has it is a female of reproducing during fertile, like between 13 and 50 years old, or maybe 45 years old. So it's like a subtype that if you have this particular genetic marker, but you have 2 copies of it, you suddenly increase risk 10 times. So it's a big effect. So we are very interested. That's a very important finding. We don't know what. We are trying to figure out how it works. It's a very strong effect. It represents about 13% of the cases in Caucasian. So about 13% of the Caucasian cases have this particular genetic factor that increase a lot of risk. And we are trying to figure out why. So to make a long story short, we sequence the whole region around it because we're trying to find if there is a mutation somewhere that explain it. And we actually didn't find anything, which I will explain later. But we at least found that, again, it's only in Caucasian. And the people who have this particular genetic factor, they have more typical disease. I mean, they basically in general, they have more seizures. Everything is more classic. So it's a very classic anti-NMDA receptor encephalitis. It also looks like in general, they relapse more. But even so they relapse more, the disease sequela, like which is the score that they have 12 months later, is less high. So maybe it's more reversible, kind of. So it looks like they have a very typical disease, but it may actually relapse more easily. But the good news is at least it reverses more easily as well. They have less sequela. So we are trying, but the key really is to figure out why, because if we could figure that out, maybe we'll find a way to treat, this disease in a very different way. And what's very particular, so when you zoom in this region where these 2 genes are, there's 2 genetic factors that are 100% together. So we cannot tell which one it is because when one person has one, it always has the other. So this genetic factor only exists in Caucasian. So you never find them in control. In African American or in Asians. So that's why it's only the Caucasians that have this form of the disease, because the genetic variant doesn't exist in other ethnic groups. So it's a subtype of the disease that only exists in Caucasian. And we're trying now to figure out how does it work, and that's where we got in trouble. Because we can't figure it out. Right now, at least we cannot figure out. We're still looking. It's such a strong effect. We have to figure it out. But basically, it's like these 2 genetic change that are in this region between 2 genes. So we suspect that one of these 3 genes is involved, either this MAD gene, this NR1H3, or this ACP2. And we're trying to study what these genetic changes could do to these genes now. That's what we are doing, but we have not a solution yet. I think one of these 3 genes must be involved, but we don't know yet which one, which is very frustrating. Now there's another gene that we found on another chromosome, which is called this DLMX2. And what's interesting about this DLMX2 is that it interacts directly with this other one that's called MAD. And MAD is this gene here. So we have 2 alternative theories now for autoimmune encephalitis. One of them is that it involves this DLMX2 MAD connection, and that really this genetic factor here is really regulating MAD. We are trying to study what these genes do on MAD, or that it's a coincidence. We don't know for sure. And that maybe there is this 2 genetic factor, but that happen to interact, but it may be by chance. And the real genes that are involved is here and here or here, plus this other one, but it's not related. I think it would be really, it's unlikely, in my opinion, because it's so much chance that this particular gene MAD directly interact with the other gene we found to be associated, that I think it's most likely a connection, but we don't understand how it works. So just to tell you what these genes do, is they are very important for an inside cells, for digesting protein or viruses, and then later on, presenting them to the rest of the immune system, something of that nature. And so we're now studying how that could work, but it's kind of early. Then there's another finding we made, which is very interesting as well, which actually was first made in China. There is this paper that was published in China. China, they have a lot of cases as well. Of course, they didn't find the ACP2 genetic variant because I told you it doesn't exist in China. So this subtype of Caucasian gene disease doesn't exist in China. But they found another gene called IF1H1 that was associated. And we also found it, the same exact effect and the exact same genetic variant. So that's another very, I think, important finding. This particular genetic variant must be also involved in predisposing to anti-NMDAR receptor encephalitis. And we don't, this is a complicated gene. It's involved in detecting viruses, double-stranded RNA. That becomes very technical, but viruses, they often have genetic material that's different from us. And they may have something that's called double-stranded RNA, because we have double-stranded DNA and single-stranded RNA. And those seems to be, so it's possible that the reason it's associated with autoimmune encephalitis could have something to do with a virus that's triggering the autoimmune encephalitis in anti-NMDAR receptor encephalitis, which, by the way, fits very well too with these other two genes. I didn't have time to really go in detail, but they're also very important for how, if a virus infects a neuron, it's very likely that these genes are important to explain how the virus affects neurons and goes from one neuron to the next. So all this seems to point out that there might be a virus connection or an infectious connection, but we still cannot find it, unfortunately, which is super frustrating. I personally believe that anti-NMDAR receptor encephalitis, there is something like a virus that we have not discovered or an infection that triggers it, and we just have to find it. But it's through this genetic factor that I think we're going to be able to maybe find it. So finally, we also are helping other people. Like, for example, the anti-GAD65 neurological symptom, I'm sure you know that's very common. And many people have this problem. It's probably one of the most common, but it's also quite complicated disease. I mean, it's not like anti-NMDAR receptor encephalitis or anti-LGI1. It's much more heterogeneous. So we are not studying this in particular, but all the samples we're collecting, we're giving them to a German group who is trying to do the same thing in this particular disease. And they are starting to find a few genes that are associated with anti-GAD65. And I hope that this will help to understand what is triggering the disease as well, and maybe also the different subtypes of anti-GAD, and maybe improve the diagnosis and try to see which subtype should be treated how. So I think that's pretty much, what I wanted to cover. I mean, I know it's very technical, but I'm going to try to summarize in a few sentences. The first one is the genetics is always causal. So that's the biggest things that you can do with genetics is when you find something, you know that it's not the disease that has caused it. You are born with it and it favors the disease. So it can give you an idea of what is the cause, not the consequence of the disease. And that's extremely important because when you really want to understand at the end how the disease is created, you need to understand the genetics. The second point is we also are finding a lot of these HLA molecules that are involved, like many of those. And this is kind of a cornerstone of how the immune system, if different people recognize different virus or yourself, to mount an immune response against yourself or viruses differently in Tabitha versus me. And by understanding this beginning, the type of HLA, the type of peptide it binds, we may be able to understand why the immune system goes wrong in some people and develop then the autoimmune encephalitis. And then the third one is there are even some that don't involve HLA, and we may discover something completely new. Like I gave you the example with NMDA receptor encephalitis. Nobody had any ideas. It's going in a completely different direction. So that's only by looking at genetics that you can do that. And I also want to thank the patients because I know I've been called, I've done that in narcolepsy, and I've been called an international vampire. And it's exactly what I am. I'm usually trying to get blood samples, add to this cohort. We publish it so everyone has access to the data. Everyone can pursue whatever we find. That's very important. Open science is critical. But that's a really good way to make progress, you know, for the whole community to do this genetic study. It's a little bit frustrating because you have to have large numbers and you have to beg, you know, but at the end you do a good thing for the entire community because anyone could pursue any of my findings and will discover something new and crack it. And then that would benefit, of course, the patients. That's the only thing that's important. So that's my conclusion, and I thank you for your attention. And I'm sorry because I know some of it was a technical.
It's okay. You can take your slides down. You're a prolific storyteller, so the fact that the first few slides weren't showing didn't impair your presentation at all. I want to make a point for everyone out there who is a patient. We have on our website under studies and clinical trials, we have the IGNITE study. Can you tell people listening how they can get involved? It's quite simple. And do they need to be in the United States, or is location important?
Yeah, unfortunately, now the regulation has become more complicated. So in general, people need to be in the US. But if someone is in a foreign country, I can probably get them in contact with someone else that can then collect a sample for us. Or someone else can do it. Because we have this international, you know, we work all together. That's why I really like actually this community. I want to say that you're not very lucky because not a lot of people work on it, but you're very lucky because the people who work on it are very good people. So that's what I've learned joining this community. So
it's an amazing frontier that you're setting out on, and it's incredibly exciting. I was thrilled to hear just what you had been able to glean with NMDA. That can be critical information for patients in the future. It's incredible.
Absolutely. This is one of the things where I think we're the most excited. And maybe tomorrow I will find out why. And I'm working many different ways to try to understand why there's this factor that increases the risk so much. Believe me, but we haven't found it yet. But I'm optimistic. And if we don't find it, we'll publish, it will be published, and someone else will find it. You know,
Right, exactly.
Yeah.
Right. Well, we'll see if we can get to a few questions. A physician is asking, they say, I'm a physician and have 3 people in my extended family with seronegative autoimmune encephalitis, confirmed inflammation, and other autoimmune sequelae, but different triggers presentations for each. Genetic exploration is always recommended, but where do families like ours start as seronegative with exploring the genetics?
That's very hard because, of course, when you're seronegative, we don't know how to classify,
That's
so
right.
it's just like only 3 cases. So if the family is big, and curiously, if the people have far, like if it's a grandson and a grand nephew or something, it's actually better because it increases the number of relationships between the people, and the probabilities that they will share something become smaller, and then actually using you can then sometimes find the cause of the disease by studying family members like that. Usually what's being done when there is a familial case like this, do what's called an exome sequencing, where you sequence all the genes and you try to see if there's anything dramatic happening in one of these genes, which could explain the problem. But very often you find nothing, unfortunately. So for this kind of families, basically you have to convince someone to sequence their exome or their genome and see if there is like a very striking difference with the control. But 3 cases is not a lot, so it makes it very unsure if you will find anything.
Now, someone else is asking, their son had genetic testing years ago. So should they recheck
No,
because, okay,
the genetics stays the same all your life. That's why.
okay.
So it's it's going to be even if you take your DNA at birth or you take your DNA at 90 years old, it's going to be the same. I mean, there are, of course, some. It's the same, basically.
And we have
I still don't really understand. I think now with the progress in technology, everyone should have his genome sequence at birth, probably because it would. But the problem is we don't understand yet all this information. You know, we can do it now. The technology is such that you can sequence an entire genome, so not even study the 1 billion markers that I said, study everything, 3 billion different genetic letters, and you can do it for about $200 or $300. The problem is nobody really knows yet how to interpret all the differences,
yes, yes.
but it will happen.
Yeah, and we have a lot of people who are asking questions who are seronegative, and I think in your studies, because this is the first study.
Yeah.
And so you're wanting people who have identified antibodies because you have to start there.
That's correct. It's I work on narcolepsy also, which is a little similar. I have a lot of time. I discovered the cause of one subset of narcolepsy, which is called narcolepsy type 1. And the reason is that it was easier to find, and there's also this narcolepsy type 2, but we don't know where to start. So for the people who are seronegative, it's a little harder because we don't really know where to start. Even if I was to find a genetic marker, what would I study an HLA? What would I study that would interact with it? It would be very difficult to figure that out. I'm not saying it shouldn't be studied. I think it should be studied, but then it's a different set of techniques. Probably the best way to study this seronegative cases is to screen, like arrays of antibodies of all the protein of the body to try to find new antibodies. But I think a lot of people are trying to do that.
Another question, and I'll make this the last question since we're out of time. And we do have several people who are thanking you for their presentation, your presentation. They have really enjoyed this. Do you see any connection between mitochondrial genetic dysfunction and autoimmune encephalitis?
That's a good question. We not obviously, but it is very possible that some of them could involve mitochondrial dysfunction. The problem of the mitochondria is it very when your mitochondria doesn't work well, it's like the factory of the cell, the energy factory of the cell. So the whole cell is dysfunctional. So it can produce symptoms that are very broad in many different tissues, similar to an autoimmune disease. So I think the mitochondrial disease and the autoimmune disease can resemble each other because they can sometimes be very broad reaching. They can attack many different organs. And it is possible that sometimes the autoimmune process can trigger some mitochondrial dysfunction. But there's no obvious, association that, in any of those diseases that we know of, at least.
Yeah. This is a good question. Someone is asking, how do AE patients contribute to these genetic pools when they don't live near a major center? How can we get you our DNA?
So that's nice. We send a kit, like
Yeah.
a saliva kit, and then you can just mail it back after you, of course, you have to discuss with our staff to make sure that you understand the study, you sign the informed consent. Of course, we want to make sure that everyone knows that this is confidential, but also that they understand what we're trying to do before they participate. But once that is done, we send a kit, you fill up a little bit of information, and that's it.
And it's a very easy kit too. You're asking for a saliva sample.
Yeah.
And the person just simply does that, takes a few minutes, puts it back in the mail to you,
Exactly.
and we're on our way. I think that was, I'm so glad that that person answered that question because we want everybody to know this is an accessible and very easy thing to participate in. So as the audience is out there, and this is going to be a very long-term study,
Exactly.
always mention if you know someone right now where we are in the frontier of this brand new area in science, if you have an identified antibody and you're talking to someone with AE, mention the IGNITE study to get your samples in so we can learn more.
Exactly, because it's also a game of number. And as I said, it's really a resource for everyone because what we find is then usable for everyone. That's very, very important to understand. Science builds on itself. So this is a kind of, it always works. Why I'm doing genetic is because it always works.
Yes.
Sooner or later, you kind of figure out what's happening. But it needs large numbers, which is very frustrating because it needs to be patient. But I found the cause of narcolepsy took me 15 years. So I think it's difficult for people to realize that research takes time. You have to be persistent, and more often you get no or no result. If you continue, and that's the way it works. You have to be dedicated, and there are certain directions it's going to work. And the technology is improving so fast now that it's very exciting that the genetics, I think all this will be, people will have their genome sequence at some point, everyone, and we'll even know more.
It's absolutely fascinating. Absolutely fascinating. I can't thank you enough for the time you've given
No,
us
it's a
today.
pleasure.
This has been a very exciting presentation, and as a patient myself, I got sick in 2013, to see how far research has come and that this whole area is now being tackled. It's incredibly exciting.
No, you're absolutely right. So often I say, like some of my friends, or they think I'm a little crazy, when we work on these things and you talk about it. But I think, like it's fantastic because not only we are helping people like you and so forth, which is very, I think it makes you feel good, of course. And then in addition, we know it's really interesting. The research is very interesting. It's frustrating because you get more often negative results than positive results. It takes a lot of persistence, but I am always excited when I find something new.
Oh, of course, you must be. You must be
Yeah,
over the moon.
exactly.
Well, thank you so much. It has been absolutely a pleasure, and we appreciate your time.
No, thank you so much for inviting me. Bye bye.
Okay, bye-bye.